Okay, I'd like to welcome everybody today to the April 13th meeting of the IMAG Working Group on Multi-Scale Modeling and Viral Pandemics. As usual, I need to remind you that this meeting is live streamed on YouTube and recorded for later viewing, so please be aware of that. We're very grateful to have multiple members of the Swedish Digital Twins Initiative speaking to us today. That's quite exciting, and we're looking forward to learning about this important program. Tom Helikar and I are co-leads of the working group. Jim Sluka and Lorenzo Vischini are also administrators for the working group. We are always here looking for your suggestions and comments for ways to make things work better. You can contact us at any time. We have our usual communication channels. They work better when you help us use them. In particular, we have our great YouTube archive. We'd love people's help in building membership and also in publicizing coming meetings. We have a Twitter handle, which is MSM Viral. If you tag things with that, it'll help us perhaps build the community. I'd like to remind you that from the 15th of May to the 2nd of June, there's the big 3-week workshop on building immune digital twins at the Institute Pascal, spearheaded by Reinhardt and Anna. If you are, many of the people in this room are going to be there. If you're still interested, please do contact them. I know there are many people here who might be interested in that. There's going to be the IMAG MSM 20th anniversary meeting in Bethesda on the 28th, 29th of June, 2023. Again, many people connected to this working group are going to be participating there. The key organizers would be Grace Peng and Herbert Sauro and Shane Pierce-Kotler. So you can contact them for additional information about that meeting. I want to remind you that here in Indiana, we still have 2 full professor positions open in the general area of biomedical digital twins. That's very broadly construed, could be anything from biosensors to machine learning for data assimilation to mechanistic modeling or physiology. If anybody's interested, we would love to have your applications. These are full professor positions with appropriate salaries and funding. I want to remind everyone also that we have GLIMPRINT, our global alliance effort, started. Tom has a membership page now. You can sign up. You can pay dues if you want to. And we would love you help us build that community. The website is active, so we very much appreciate that. A couple of things have come across my desk just in the past few days. There's a Lorentz Center eScience workshop competition. And we've been thinking about the possibility of trying again for that. It's specifically on computational mathematics for real-world applications. And so some of the technical challenges that would be involved in building medical digital twins, how do you do parameter identification? How do you do inverse problems? How do you do model updating might be of interest. So if anybody is interested in putting in a proposal, we need a 1-page proposal by April 25th. That's coming up very soon. But I'd be happy to talk to people if they're interested in that one. Another thing that came up that might be of relevance to the community with another very tight deadline is that the Wellcome Trust has a Wellcome Data Science Ideathon competition. And one of the areas that they're specifically interested in is improving treatment for infectious disease. And so it occurred to me that the immune digital twin concepts might be of relevance to those. They prefer that the lead for that be in the UK. Lorenzo, I'm thinking of you perhaps. And so it's possible that that could be of interest to people. If they approve those meetings, those will be quite soon. Those would be in July of 2023. So if somebody wanted to do something after Paris, that might be a possibility. We will have a steering committee meeting tomorrow at 11 a.m. If you need access and you don't have it, please contact me, Lorenzo, Jim Sluka, or Tom, and we'll send you the Zoom links. Are there any other announcements before we move on to the speakers? No. Okay, well then I should say that coming up, we have Claudio Montero from Nova Discovery talking about their great software platform for the second time in 2 weeks. May 11th, we have David Basanta, who's done wonderful work on developing improved cancer therapies using patient-specific mechanistic models. And then May 25th, we will not be having a meeting because of the Paris workshop. And then we'll be having Megan Hayes from University of Virginia talking about her work on modeling a muscle regeneration in biomedical contexts. If you have suggestions for speakers, please let us know, and we will do our best to get them on the agenda. As always, Lorenzo is extremely good at getting a schedule put together. But again, no meeting on May 25th because of the Paris workshop. So I don't want to take any more time away from our speakers. It's already late for them in Sweden. We really appreciate you being willing to speak to us, and we will turn over the meeting to you.
Thanks very much. So Martin, could you share our slides, please? So I'll talk about digital twins for predictive, preventive, and personalized medicine. And this is a team effort, so we will present it as a team. And next slide, please. So here is a capsule summary of what we want to do. To the left, you see a patient with a local disease, and you computationally construct thousands of digital twins of that patient's based on multiple different measurements from the patient. And those twins, are computationally treated with thousands of drugs to find the drug that cures the twin. And that's the green symbol. And that drug is given to the patient, who ideally is cured. Next slide. And why do we need digital twins? Well, it's a staggering number of patients that are not really cured by, or at least the medication is ineffective in a large number of patients with complex and malignant diseases. And an important reason may be that lots of medical research is based on, next slide, this assumption that you can find and target an upstream gene in a disease-associated pathway. Like in A, this symbolizes a disease-associated pathway. And if you target the upstream gene with B, the blue drug, then you can ideally silence the pathway and cure the patient. But next slide. In reality, of course, each disease usually involves hundreds or thousands of genes and many pathways, so that it's just not enough to find one upstream gene. But as you can see here, there's a potential structure in this, which is network modules, which is what we use as the framework to construct digital twins using network modules. And the idea here is that you have the protein interaction network, which here is symbolized by white dots that are connected by predicted interactions. And you computationally match blue disease-associated genes on that network. And the most important disease genes tend to co-localize and form modules, and this is where you find biomarkers and drug targets. And we have shown that this actually is an effective way to find biomarkers. That's almost 10 years ago. And next slide. An important advantage is that you can build multi-layer modules starting with DNA to construct high-resolution multi-layer modules. And this principle can be extended to symptoms, which also can be described by networks. So we have something like 500-600 symptoms in human diseases, and they tend to be also co-localized in such a network, so that you can connect a module of symptoms corresponding to disease to their underlying molecular modules. And you can, next slide, continue to construct high-resolution multi-layer disease modules by adding network modules of lifestyles and environmental factors. And then you can use machine learning to extract combinations of predictive variables for different types of personalized drugs. And next slide. And this all sounds very simple, but there are important problems to solve. And one is that there are different types of molecules like metabolites and genetic variants. And Dina Mansour Ali and Firoz Mahmoud will talk about this. And two, involvement of thousands of genes across multiple cell types. And Yelin Chao will talk about this. Okay, you went a bit quickly here. And those genes differ between organs, Martin Schmelik. And the genes differ between time points, Shinshir Li. And the genes may differ between patients with the same diagnosis. And Samuel Schäfer has worked a lot with this, but it will be presented by Martin. And another important part is to transfer information from the huge public data sources into digital twins. And this will be presented by Martin too, because Oleg Sisev could not join us. And finally, I will talk about the wide gap between this complexity and modern health care. So if we start with genetic variants, Dina Mansour Ali, please.
Thank you, Michael. So I will talk about the involvement of genetic variants in the construction of digital twins. Next slide, please. So when I say genetic variants, I'm referring to single nucleotide polymorphism. These are the SNPs that are identified by association analysis between the genotype and phenotype of interest. The phenotype is a trait of which are interested in properly the disease. In a genome-wide association study analysis, and usually these SNPs are published at GWAS catalog. Next slide. So one way to use a genetic variance is to construct polygenic risk scores. A polygenic risk score is an individual personalized score for each individual. It is constructed using the sum of all the SNPs weighted by the effect size that's carried by that individual. And we use the SNPs that are associated with certain disease of our empire, play a very valuable tool that enable person assessment for a risk for certain disease. Next slide. Yes. So in our current project about pancreatic cancer, we have a shared multicellular model based on single-cell transcriptomic data. Differential expressed genes, the DEGs, per cell type are going to be processed in two ways. The first method will be GWAS enrichment to determine genetic variants associated with these differentially expressed genes. And the second one will be metabolic enrichment, which will determine the metabolites. My colleague Firoz is going to talk about the metabolic enrichment. From my side, the GWAS enrichment will be used to identify genetic variants that will construct polygenic risk scores to diagnose pancreatic cancer, prognose pancreatic cancer, and early risk prediction. For the diagnostic PRSs, we are going to use the genetic variants that map to the pancreatic cancer GWAS. So any genetic variant mapped to pancreatic cancer will be used for diagnosis. Any genetic variant that map to any cancer-related trait, like survival, tumor size, metastasis, and extra, will be used for prognosis to determine the performance of the patient during the course of cancer. And the third one would be the variants that map to any pancreatic-related trait, like pancreatitis or type 2 diabetes, and these will be used for early prediction of the risk of pancreatic cancer. This is all from my side, and I will let Ferouz talk about the metabolic enrichment.
Thank you, Tina. I work with the relation between metabolites and other omic layers like gene and its involvement of metabolites for disease progressions. Next slide. So what is metabolites? As we know, the metabolites is the end product of transcription, translation, and so metabolites is works as the energy and building blocks of the all life. So metabolites is important for to connect the disease and the disease states. Next slide. So normally when we have different conditions, like here is cancer and healthy patients, we can extract the metabolites information by mass spectrometry, and with the help of multivariate statistics or machine learning, we can find out the metabolites which are differentially enriched in a certain states. Here, in case pancreatic cancer, as an example. So once we have the metabolites, we can connect the metabolites with the gene information which are differentially expressed in a state, and also we can find out the metabolites a pathway which is overlapping with the gene expressions. So in that sense, we can narrow down the mechanism which is very much responsible for a disease progressions. Next slide. So not only that, if we have metabolomics data, then we can find out the information of the disease. But if we don't have the metabolomics data, we can still create the genomic scale metabolic model. Here you can see that we can create healthy, mild or severe cancer patient with only the gene expression data sets. And once we have the gene expression of a certain state, we can connect the metabolites and find out what is going to happen narrow down in the mechanisms. And that's how we can connect the gene with the metabolites. And the thing is that is bulk. It's not as straightforward as it looks, because we have multiple organs, and every organs has multiple cells like T cells, B cells, or other immune cells. And so single cell levels, we need to have more layers. And that's my colleague Yelin is going to take what is more complexity.
Thank you, Feroz. I'm a PhD student, mainly work with analyzing single-cell sequencing data and creating multicellular disease model of different cell types. So next slide, please. As shown in this slide, each disease, we can see that involves many, many different cell types. If we study the disease at a bulk level, then we miss a lot of valuable information about how each individual cell type performs. That's why we use single-cell sequencing technology to characterize each cell type in the disease. So by performing single-cell RNA sequencing analysis, we can dissect the whole tissue to a cellular level, allowing us to distinguish between different cell types and identify potential treatments that are targeting certain specific cells. Next slide, please. Using this method, we found that there are thousands of genes have altered expression levels in different cell types. Then the question comes, how do we identify the most important genes and cell types? To do this, we developed a strategy for identifying the hierarchy of genes and cell types. So what we do is through the bioinformatic analysis, we traced upstream regulators and upstream cell types. So as shown in figure A, we have different differentially expressed genes, which show as red dots in one cell type. So in figure B, we identify the possible upstream regulators of those differential expressed genes in this cell type. Then goes to figure C, we locate the cell type that contains these upstream regulators. So by doing this, we are able to create a cell-cell interaction model, which we refer to as the multicellular disease model. Next slide, please. This one. Using this method, we constructed the multicellular models of 5 different cancers. As we show here, each dot on the multicellular model represents a cell type, and the lines indicates the interaction between each pair of cell types. As you can see from this slide, all cell types are highly interconnected, and as no clear upstream cell type can pop up, and genes cannot be identified from any of this model. Because the similar complexity across different cancers, then we reason that we could make a shared multicellular disease model across different cancers and use this model and the network tools to prioritize genes and cell types that may be important in multiple cancers. The prioritized genes might be suitable for diagnosis and prognosis. So as shown from the left figure, we extract common interactions from the 5 different cancers and created a shared multicellular disease model that represents the interactions that are common to all 5 cancers. Then in figure B, we use this shared model, prioritize cell types and upstream regulators, and identify possible candidate biomarkers. Those biomarkers then validated for both diagnostic and prognostic purposes. For diagnosis, we tested RNA expression in 27 different cancers and protein expression in 9 cancers, and all of which demonstrate good predictive abilities. For prognosis, we validated in 23 cancers and found that the combination of candidate biomarkers could predict survival successfully. Moreover, we found that in an independent liver cancer dataset, the polygenic risk score, which Dina developed based on the shared multicellular disease model, were higher in severe liver cancer patients compared to less severe liver cancer. This study is currently under review. And next slide, please. This concept of using multicellular disease model for identifying biomarkers is not limited to only cancers. It can also be applied to immune diseases such as arthritis. The same, from the multicellular disease model, we found that it shows complex intercellular interaction, and there is no obvious top upstream cell types or genes can be identified. So we used the same network tool to prioritize cell types and genes. In this paper, next slide, please. The selected top upstream regulators were chosen as treatment target and therefore are validated in a mouse model. The treated mice has the lower arthritis severe score compared to untreated mice. So to sum up this part, we studied different cell types. However, a disease is not only involve many cell types, but also many organs. Therefore, Martin Smelek will present his finding on multi-organ studies next. Thank you.
Thank you, Yelin. So my name is Martin. I'm a PhD student in Mikael's group. And in this part, I will talk about 2 projects. In the first project, we analyzed PBMC spleen and joint data from a mouse model of arthritis. And we basically wanted to explore a simple idea, which is that given that inflammation is active mostly in joint, and it is complicated to get samples from it, we wanted to search for shared biological mechanisms between PBMC, joint, and spleen. And ideally, these biological mechanisms would correspond to the disease progression. And the idea is to later measure biomarkers for these biological mechanisms in PBMC and therefore avoid getting samples from joints.
Sorry, Martin, maybe you need to say what PBMC is.
Oh, yeah, sorry. PBMC, it's a part of a blood that corresponds to the immune cells, basically. Yeah, so figure on the right side shows these like 15 different factors that we computed, and their estimated expressions in joint PBMC and spleen. So for example, we can see that factor 9 has a high expression of, factor 9 is highly expressed in PBMC. So for these factors, we recovered, or we searched for their biological mechanisms and like what to what biological mechanisms they corresponded. And we found out that mainly factor 6 was very important in the disease progression. And this one corresponds to the interaction of humoral and innate immune response. And we later found biomarkers representing these factors, and they were validated in independent human dataset. And in this project, we are now in the stage of kind of finalizing the manuscript. So then there is a second project I would like to talk about, which was recently published in Cell Reports Medicine, where we analyzed multiple organs from mouse model of arthritis. And interestingly, we found out that not only joint, but many other tissues have differences between healthy and sick. And in the downstream analysis, we focused only on joint, lung, muscle, skin, and spleen. And we constructed this multi-organ, multicellular disease model that you can see on the figure. And on the figure, each line represents an interaction. And the figure basically shows how highly interconnected different cell types are, and how highly the organs are also interconnected. So with this, we moved on with the second part of the project, where we try to understand what kind of different disease mechanisms there are in different organs. And we organized more than 400 pathways into different transcriptional programs, and we identified program 1.6 as the most important. And this program was full of inflammatory pathways and stress-related pathways, which makes a lot of biological sense. And they were downregulated in muscle, but upregulated in joint. And this suggested something that we call then a graded on-off switch, which shortly means that instead of one gene being a driver of the disease, there are several genes with interconnected effects that can be gradually off balance. And depending on how much off balance the genes are, the more serious the disease can get. And as we see, this switch is turned, this switch is basically turned off in the non-inflamed tissues, and it's turned on in inflamed tissues. So we propose that such upstream regulators of some such programs might be promising therapeutic targets. So, but as we know from the clinical experience, the disease changes over time. And this is what Xinshu Li will present in the next part, where she will talk about how we integrated the time variable into our analysis.
Thank you, Martin. I'm Xinshu Li. Here I will talk about can we find the early upstream regulator. Next slide, please. In one of our early projects, gene regulatory network, we constructed by the time series profiling of the transcriptomics of human CDT cells. And we found that early regulators of disease can regulate the early and late stage target. But there's a problem for the human disease. It's difficult to get samples before patients have the symptoms. Therefore, we analyzed the seasonal allergic rhinitis. We mimicked the disease process by challenging the PBMC from patients with this disease and performed the time series analysis to identify the early events. Next slide, please. We use the method Yelin mentioned before, based on the upstream regulator and then downstream target to construct the MCDM for each time point. You can see all the time point, all the cell types were highly connected. So there's no early upstream cell types, and there even no one single upstream regulator. Instead, we ranked the upstream regulators to identify the upstream regulator that had the more target and more time points. Next slide, please. In the left figure, you can see I4 as a drug target for the seasonal allergic rhinitis can only regulate a few cell types and a few time points, but PDGF-beta as a top upstream regulator can regulate most of cell types and most time points. The next layer of the complexity could be that not only genes differ between different time points in the same individual, but they can also differ between different patients and between the same diagnosis. Martin will present how that will be exploited for personalized medicine. Thank you.
Yes, thank you. So this is mostly work done by Samuel Scheffer, but he couldn't join us, so I'll present instead. And in this project, the aim was to predict a personalized treatment based on the network models. And the key idea of the project is that if we map disease-related protein, shown in blue on the figure, into protein-protein interaction network, they tend to form modules, as Mikael explained in the beginning. And the approach of this project is about localizing these modules and optimizing which drug targets would treat the module in the most efficient way which corresponds to the red thingy on the figure. So very shortly, what Samuel did is that he constructed a network of interactions between different cell types, as well as a network of interactions within each cell type. And then for each cell type, he found a disease-related module in the network. And based on the cell type importance, he computed a ranking of thousands of drugs from a drug bank to find out which is the most important, or which is predicted to be the most important. So then he extensively validated the rankings in independent human data sets, as well as in vivo and in vitro mouse models. And when validating in human data sets, he realized how much drug ranking changes based on the inter-individual differences between people, which is shown in this figure, where we show two patients that have the same disease, which is Crohn's disease. And the patient on the left has more kind of B-cell heavy progression of the disease, which concluded with a completely different drug ranking, where more alternative drugs rank the best compared to the patient on the right, which has more stroma cell and T-cell heavy progression, and in which the approved treatment ranked much higher. So this project highlights why the granularity of single-cell RNA is important for the drug prediction. However, there is already a lot of known knowledge from bulk data sets that would be great to use as well. And this is something that Oleg works in our team on, but he cannot join, so I will continue talking. So, so the digital twins for drug prediction, there is the alternative A, which is basically to use the multicellular disease models and look at the targeted proteins from the drug bank and do drug prioritization based on this, which is basically what I just shown, which was Samuel's work. But we also have large bulk drug trials measuring omics and drug sensitivities. And we have more detailed drug info, which is chemical structures, side effects, and much more. And if we do alternative A, we throw away all this important information. So what he's developing is basically a transfer learning approach, where he tries to transfer information between similar domains, which is single cell RNA and bulk RNA in our case. It's just the concept of transfer learning is that if you train a classifier on like differentiating dogs and cats, you can also transfer this knowledge into differentiating then the cartoon images of dogs and cats. So this is in a nutshell what he proposes, or what he's working on, where the input data would be the single cell data from the patients, and the bulk data would be the bulk data from the repositories and from the public domain. And as well, we would have the drug properties with all the chemical structures and much more. And basically all of this would be, for all of these neural networks would be used together with transfer learning to predict the drug effect of the on the single cell level, but using all the information from the bulk level. So I'll give the last word to Mikael.
Thanks. I love having the last word. These were snapshots of what we're doing, and the background to the very short presentations is that each project deals with incredibly complex data and methods, so it would just not be possible to present it in detail. But next slide. We believe that this is doable, and for example, Samuel's method could be applied now to individual patients that don't respond to treatment. And we believe that the ongoing efforts in this field will reach the clinic within the next few years. And next slide. Or if you're interested in more information about our work, you can find it on our website, sdtc.sc, which includes publications and other more detailed information. Next slide. And of course, many people are involved, so I'd like to thank them. And finally, of course, our funders. So thanks very much.
Thank you for the great presentation. Can I ask, I'll open the floor to questions. I'll put the summary slide, the abstract slide back up. But if you need to take the screen back to answer questions, that's fine. So the floor is open for questions. If somebody has a question and you can't unmute because you don't have permission, just put it in the chat and we'll make sure you get the question. That's an incredible project that you've developed over the past many years. I had a practical question, perhaps, which is a lot of the data sets that you're looking at are ex vivo. Mm-hmm. And so I was wondering if when you start translating to humans, do you do the pathway construction in mice and then use human blood-derived data sets, or some of these things were clearly more medically directed than others? But I was wondering if you could come back, and your last slides were about translating this to medicine. I was wondering about coming back to this question of how much of this is usable, can be done using easily clinically accessible data sets.
Well, that's of course a key question. We use mouse models to find overriding systems level principles like multi-organ, multicellular disease models, which is not possible in human subjects because people are unwilling to give samples from their brains or liver or heart. But we are translating those, like in the last Cell Reports Medicine article, we started with a mouse model to construct a multi-organ model from which we identified transcriptional programs, which we verified in studies of 11 different autoimmune diseases. And we also compared with, we analyzed blood samples from 600 patients with SLE. And those blood samples were used to analyze upstream regulators of the transcriptional programs. So yeah, so the essence is that mouse models are used to identify overriding principles, and then we check if they are useful in clinical settings.
That makes a lot of sense. Clearly doing multi-organ sampling is going to be tough in humans. And so coming from mice seems to be a great approach.
So I have a question, if I can ask. So it's amazing work, a lot of different interactions and models. It's absolutely wonderful. I think I would love to hear each one of the students give a longer talk. But so I know that I think a couple of students talked about the cell-cell interactions. And I think one of the study was from PBMCs, and if we take that as an example and think about the different cell types, so there is definitely inherent directionality in the way cells interact, right? So for instance, the innate immune cells would lead to T cell, and then the T cells would lead to activation of B cells. So are your models taking this inherent directionality that we know in the immune cells in account, or that's not taken into account and you allow all sort of cell-cell interactions?
Yeah, since all the immune cells are not excluded from all other cell types in vivo. Immune cells interact with stroma and other cell types, many of which have immune functions. So you can't really separate different types of, you can't separate the immune system from the rest of cell types, and you can't separate different types of immune cells from each other because they all interact, at least in our studies. And when it comes to directionality, yes, the idea is that the innate system comes first and then activates adaptive mechanisms, but that is very hard to study in vivo in human subjects because it's very difficult to find the initiating event in a patient because symptoms occur much later. But as shown by Xinshu Li in her studies of allergic patients, we could not find such a hierarchy where even at an early stage in the time series analysis. So we analyzed symptom-free patients with allergic rhinitis during winter when they were not exposed to pollen in vivo. We exposed the PBMC to pollen, and even before pollen, all the cells interacted in a way that differed compared to healthy controls. And there was no, after pollen challenge in vitro, there was no clear hierarchy between the cells. But of course, these cells have been sensitized at some previous time point, maybe years before our in vitro challenge. So it's difficult to, it's the in vivo complexity is different from that in textbooks, models.
Yeah, absolutely. I was not thinking about the tissue specific or organ specific models, more in terms of PBMCs when you would mostly see only immune cells. But it's interesting that that hierarchy is not observed. Thank you.
Actually, maybe I was too black and white there. I mean, there is a hierarchy. Some are more important than others, but there is no clear linear hierarchy. So it's a complex subject to discuss.
Yeah, but at least, I mean, we can talk about this, but T cells, right? The T helper cells are going to induce the B cells. So at least if that level of granularity, I wonder if you see in your model. Yeah.
But that it's very difficult because this in vivo in patients, they are already sensitized even before allergen challenge in vitro. B cells and T cells interact in different ways compared to healthy control. So there is no obvious T cells comes before B cells.
Yeah. True. Yes. Yeah. Thank you.
Other questions?
We could talk a lot about this.
That's what I was thinking actually. So I think the early, I think infant immune system is probably where one could look at, where you actually have the infant who's born and so have the placental samples. And now actually we have some of these placental samples and then early immune time points. So it would be interesting if that hierarchy can be observed in those when we know the time of challenge. But absolutely, I agree. Yeah.
That does sound very interesting.
I have a quick question. Well, perhaps quick. You've been looking into like drug bank and other drug resources, trying to find a drug that would interfere with a particular place that you seem most likely to have an effect. Have you given any thought to just identifying the point that seems most likely to be interferable? If we had a drug, or if we had, imaginary drugs for this particular individual at this particular date and time, we want to hit this gene right here, or this gene product. Drugs don't exist for that, so that means we should try to, develop that drug. Genomics has been around for pushing 40 years now, and it has really not been terribly helpful in defining where you really want to interfere in a disease. That's why 90% of new drugs fail, is because it turns out that target doesn't actually do what you want it to do. So any thoughts on using your tools to identify druggable targets?
I think that's Martin, or isn't that more something close to what you were?
I can try to answer.
And maybe Shinshu too.
Very good question. And I would say that, so there are two different approaches. One is what Samuel did with the drug bank, which has advantages. Then to what you're suggested, we kind of believe at this point that it's not that I'm not really convinced that it's possible to find one drug target. I actually believe that it should be kind of a combination of multiple drug targets that should be kind of treated in a combination. So this is something that I try to explain as this graded on-off switch. So yes, we in the results of our methods, we have a suggestion for which kind of upstream regulator would be the best to treat without looking at what kind of drugs are available for it. But at the same time, I think it would be better to, and this is what we are now searching for, actually, is a combinatorial treatment where we would choose two different upstream regulators, where each upstream regulator would target kind of a different part of the disease modules, or like disease parts, so that they could work together on treating the disease kind of as efficient as possible. So we are working on it, but still there's a long way to go, I would say.
Maybe Shinshu's So I think to some extent, it boils down to a very basic question that even if we can't find an upstream, a captain or a general cell type or gene, we can rank cell types and genes based on their relative importance. And Shishu, would you like to maybe comment on the slide that you showed with IL-4 and PGF?
Yeah, if it's difficult to find the one, it's upstream regulator.
Maybe I can share the slide. I'm sorry. Maybe I can share the slide so that everyone see what we're talking about.
Yep. Thank you, Martin.
Oh, I just need to find it.
You can see it's the left side. It's usually it's IL-4. It's at the drug target, but you can see it's not all can be the regulator all the cell types. You can see it's some may be regulated, but it's not significant. And the right side is the PDGF beta. You can say it's mostly of cell type. It's relative, but it's not all the cell type. So it's difficult to find this one. It's a perfect one. You can think that it's the perfect drug target can cure everything. So now it's just like Martin said, we would like to find it one is maybe target most of cell type or most relative cell type, and then we will find another one. It's also combined together. Maybe it's have a good chance to cure this disease. So we are working on this one now. Yeah, this is from me. Thank you.
Lorenzo?
Yeah. First of all, thank you for the talk. It was really amazing. I have one. I'm not up to date with the latest on analysis of genomic data, but the latest that I read were about finding some best candidate genes, top candidate genes, and so on and so forth. So connecting to the first slide that you presented where you show a network, and it ends up that probably there are elements of small world where at the end everything is connected with everything. Is there any news, let's say, some comments that you make on some technology that you are developing to go out of the candidate gene logic and perhaps creating something that can give us some more mechanistic insight for like one gene follows the other and so on and so forth, and actually determining the laws, the kinetic laws about that regulate those?
That's a really complicated question. So we showed two possible approaches. One is to construct gene regulatory networks where you attempt to identify computationally or bioinformatically early upstream regulators. And another is what Martin showed, which I think is very promising, is to reduce the enormously complex gene expression changes into transcriptional programs and identify the upstream regulators of those programs. And it turns out that many of those upstream regulators are already known drug targets or candidate drug targets. So those are promising avenues to find drug targets based on functional understanding of their understanding and prioritization of their mechanisms.
Thank you.
Tom? Go ahead.
So I should also say, Dina, would you like to comment on the potential of genes to or genetic variants to prioritize mechanisms?
Yes, I would like
Very
to
complicated question, but
thank you so much. So if we take a look at the genetic variants identified by the GWAS, we usually, the genetic variants first are identified from a large sample size data set. It's a population genetic studies, genome-wide association studies. So here we have a very large sample size of the trait of interest, as well as if we think that these genetic variants are the first or early stages, so the mutation or the change in the DNA starts first before any of the cascade of the disease. I think so these genetic variants, when weighted by their effect size from the GWASs to construct the polygenic risk score, which is a personalized score for each individual, can be helpful to identify the group of individuals at high risk. For example, when we presented our presentation, we had a prediction risk score for pancreatic cancer. For this, the prediction one, we use the genetic variants that map to pancreatic-related traits. So we assume that pancreatitis, for example, some of the people who have early pancreatitis could proceed later into a pancreatic cancer. How much is the risk? Does everyone suffer from pancreatitis has the same risk to develop pancreatic cancer? Of course not, because this will depend on the genetic load of these variants weighted by their effect size in different individuals. So we can have two individuals with pancreatitis, yet one has a very high risk to develop pancreatic cancer and the other one has a lower risk. By identifying the higher risk earlier, we can intervene using different interventions, like, for example, treat the pancreatitis. If we are, for example, also the risk factor here is type 2 diabetes, we can do another therapeutics earlier before the cascade develop to pancreatic cancer, for example. So this is the beneficial effect and the valuable tool of genetic variants in prediction of early disease. Thank you.
Tom Helikar had to go for a family situation, but he wrote, he had a question he'd hoped to ask, which was, would you be able to see more hierarchy potentially if you included more detailed consideration of causality and mechanism in your data analysis?
But we do, I mean, we are talking about thousands of genes that vary across many different cell types, and we organize those genes based on known protein interactions. So this is a mechanistic approach. So I think we already do this.
Okay. I'm not here to. Unfortunately, he wasn't able to ask the question himself, so I'm only relaying it. I think perhaps the focus of the question was more on the timescales over which you're looking. You either have ex vivo or long interval time series, and I think Tom in particular is interested in its short-term dynamics. How does the immune response change over periods of minutes, hours, days? And so I think of course, they're different. You see different things when you look on those different scales.
Yeah. Maybe Xinshu, it's difficult to say. I mean, Xinshu studied allergen challenged PBMC over a week. It's difficult. Can you answer that question, Xinshu?
Yeah, it's difficult to answer, and it's I think I don't have a really good answer now.
It's difficult because there were thousands of genes that changed at each time point, and many of them differed between time points.
Yeah, it's also it's in the real life, it's different with the way we use the library work, and its time is different. Also, it's like Dina said, it's a genome virus. It's also different. It's for each individual. So it's I think it's complicated to have a good answer now.
Maybe I could also just quickly add that the noise of the data it's a lot of complications if we wanted to do this approach, because the methods still are not perfect and there's a lot of noise. So we cannot really like do 2 experiments an hour from each other and like look at what has changed, because there is like so much noise that this would, I mean, it's then like a complex mathematical problem to solve.
I think I put in the chat, we had a talk by Rita de Almeida about a year ago, and she does the same kind of functional clustering that you're talking about. And she's able to show that in those cases, if you do the functional clustering correctly, you can get aggregate signals that are meaningful over a period of hours. It changes the gene expression. And so while you can't do it for individual genes, once you cluster them the way you talk about, even very short time changes are discernible. And so I put that in the chat because it seemed to me to be a method that is not as sophisticated perhaps as what you're doing, but could be perhaps complementary to some of the approaches you're taking. So great. Any last questions? Our speakers have been very generous in staying up so late.
Well, thanks very much. These questions have been very interesting to us and helpful to us too.
Well, thank you then again for coming to speak to us. We really appreciate it. It was very stimulating. I also appreciated the very excellent handoff for your large group. The polish of the you talked about interconnectivity. The interconnectivity of your group really comes across very vividly in the way you present.
Thanks very much.
We should probably encourage more groups to try to do this kind of a collective presentation. I thought it worked very well. I appreciated it.
Thank you.
Okay, I'd like to thank everybody for coming this week. We'll see you in 2 weeks. And I'll see many of you in Paris after that. Thanks again. Bye.
May I take a second to ask a question as an outsider?
Please go ahead.
It is, do I understand that we are talking around expanding like drug discovery from targets? Are we doing a bit of inverse process here, that we have some identified diseases and now we want to refine via network technology, mathematics, statistics, and data analysis to go back and refine the wild drug discovery, which until not so far in the past was just guesswork, wild guesswork and exhaustive search. Am I very far in my asking this kind of question?
Well, then I think that's a very positive way of, I mean, this is how we feel, and thanks for that comment. That's how we and others are trying to contribute to drug discovery.
Yeah, because yes. Lorenzo's comment gave me the guts to ask this question. If everything is connected to everything, one can use data to build all kinds of networks, even imaginary or hallucinating type, and eventually something will emerge.
And that's why it's important to validate with independent studies.
For sure, yeah. Thank you.
Well, thank you, Itamar, for asking. And I thought that was a great way to sum up the meeting. So with that, I will call the meeting to a close and thank everyone for participating.
Thank you.
Look forward to seeing you again soon.